Okay, children. Good morning. Today's topic is ABO blood types. Okay, beta. ABO blood types. Okay. So this ABO blood group typing is explained by or discovered by Carl Landstein. Carl Landsteiner. Okay. He explained about the ABO blood group system. He explained about ABO blood group system. So here he was awarded the Nobel Prize in 1930. 1930, he was awarded the Nobel Prize for his works in physiology and medicine. For his works in physiology and medicine, he was awarded the Nobel Prize in 1930. So let us see here three alleles according to the concept of multiple allelism in blood group system A, B, O. Here these are three alleles, and here phenotypes. Phenotypes A, B, AB, and O. These are phenotypes. Okay. Next, later we will discuss about the genotypes of these phenotypes. Okay. First of all, here how these blood groups are classified into A and B and ABO. So here the type of blood groups are characteristics of their presence of or absence of antigens on the plasma membrane of RBC. See beta here, this is RBC. Okay, so this is the plasma membrane. Plasma membrane. So on this plasma membrane, presence or absence of antigens. Antigens. Basically, these antigens are carbohydrate groups. Carbohydrate groups. These antigens are carbohydrate groups or synthetic polymers or sugar polymers. Sugar polymers. Okay, these are sugar polymers. So these antigens are present in the plasma membrane of RBC. Okay, just we can see under a electron microscope a small pimple-like structures protrudes means small pimple-like structures we can observe here. These are antigens. Okay, here these antigens can form clot with another blood type. For example, this is A blood group. If we transmit B blood group, here these antigens causes clots. Okay, so this kind of transmission of blood is called incompatible transmission of blood. Okay, beta. For example, here antigen A is there. Okay, this is belongs to blood group A. If antigen B is there, this blood group is belongs to B group. If both A and B antigens are there, here this group is belongs to AB blood group. If there is no antigens, it is belongs to O blood group. Okay, okay. So in this way here, blood groups are divided or classified. Okay, if A antigen is there, that is belongs to A blood group. If B antigen is there, that is belongs to B. If both AB antigens are there, this group is belongs to AB blood group. Without antigens, O group, right? Okay, I will show you. Next, in the plasma of blood, antibodies will be there. antibodies. Okay, these antibodies are basically belongs to IgM type of antibodies. IgM type of antibodies. Okay, so let us see here if we observe these antigens. Basically, antigens are called isoagglutinogens. Isoagglutinogens, antigens are basically called, and antibodies are called isoagglutinins. Isoagglutinins. Okay, isoagglutinins, or another name also there, isohemoagglutinins. Another name, isoagglutinins. Okay, the antibodies, these are discovered by Bernstein, but here these antigens are explained by Carl Landsteiner, right? Say antigens are called isoagglutinins, antibodies are called isoagglutinins. Here gens and the nines, please remember this pronunciation, isoagglutinogens, isoagglutinins, or hemoisoagglutinins, right? So based on the presence and absence of these antigens, and based on the type of antibodies in the plasma of blood, here these blood groups are classified into A and the B and the AB and the O blood group. Okay, let us see better here. I will show you. Here, see, this is blood group. Okay, antigen, antibodies. Okay, here A group, blood group A, here antigens on the plasma membrane of RBC. Here A type of antigens are present, and antibodies in the plasma. Antibodies in the plasma, here B type of antibodies will be there. A type. Next, if we take B blood group, in this group, here B type of antigens will be there, and antibodies, A type of antibodies will be there. Okay, please observe carefully. It is very simple concept. And if you take AB blood group, here both A and B type of antigens will be there on the plasma membrane of the RBC, and in the plasma of the blood, here there is no antibodies. Antibodies are absent in the AB blood group. Next, if we take O blood group, here antibodies are absent, and here A and B, both type of antibodies will be there. Okay, so here this is the blood grouping based on the presence of antigens on plasma membrane, and based on the presence or absence of antibodies in the blood plasma. Okay, okay, but here this is ABO blood types, right? Here this is, these antigens will present on the plasma membrane of RBC. These antibodies are present in the plasma of the blood. Okay, here these are IgM type of antibodies. These antibodies are IgM M type of antibodies. Do you know what is the importance of IgM? It is largest or smallest? Here it is largest antibody among all 5 types of antibodies. Okay, so here this is about ABO blood types. Next, here these alleles, which alleles? A, B, O. These 3 alleles are located on the homologous chromosomes. Okay, number 1, sorry, 9 pair, chromosome 9th pair. Which pair? On the chromosome 9th pair. Here these alleles are located. So these 3 alleles will exist in populations, not in individuals. Even this blood group also. Some people are with A blood group, some people are with B blood group, some people are with A, B, some are with O. Like so, these blood groups exist in the population, not in an individual. Okay, right. This is about ABO blood types. So these blood types are explained by Carl Landsteiner in 1930. Next, another scientist, Bernstein is there. He explained about genotypes. This scientist explained about genotypes. He observed genotypes of AB and AB and O, 4 types of blood groups. Here, Bernstein explained about genotypes of ABO blood groups. See, O blood groups. Blood groups. See, better here he named as I for this gene. I. The gene which is responsible our blood groups is named as I. Okay. This gene is located. These genes are located on the 9th pair of our chromosomes. Next, okay, for example, here A blood group is there, IA. This B blood group, IB. Okay, right. Here I for O. This is O. Okay. So this gene produces this allele produces A type of antigens. Please remember this A allele produces A type of antigens. This B type of allele produces B type of antigens. But here small I, this type of, it is indicated like this or like this. Okay. So it does not produce any antigen, right? So if you take genotypes of ABO blood groups, here A blood group genotype is IA, 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 I0. This is A blood group genotype. Next, if you take B blood group genotype, B blood group genotype, IB, 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 I0. And if we take AB blood group genotype, IA, IB. Okay, you take O blood group genotype, okay, like this or like this. Okay, so these are the genotypes. Next here, IA, IB, IA and IB, these are dominant alleles. And here IA, IB both are dominant and B, but these two are codominant on each other. These are codominant on each other. Next, here IO, it is not dominant beta, it is completely recessive. Say we can indicate these like this: IA is equal to IB. Both are equal, both are dominant, but both together produce codominant trait. But here these are greater than IO. Okay, please remember this equation or this thing. IA, IB both are equal and dominant, but IA, IB both together forms codominant trait. Okay, but A is greater than I, and as well as B also greater than I, right? So please remember this one. Okay. See, beta, here these are genotypes. How many genotypes are there? 1, 2, 3, 4, 5, 6. Okay. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. See, as per our formula, here 3 means n into n plus 1 by 2. Here n is equal to number of alleles. How many number of alleles are there here? A, B, O. These three are alleles. Okay, now. So if we apply this 3 number here, 3 into 3 plus 1 by 2 is equal to 12 by 2 is equal to 6. Here number of genotypes are 6. Number of genotypes are 6. Okay, this is A blood group genotype, this is B blood group genotype, and this is AB blood group genotype, and here this is O blood group genotype. Okay, so with these 3 alleles, here 6 genotypes are possible. At the same time, here 4 phenotypes. These are 4 phenotypes. Okay, here 3 alleles, 4 phenotypes, and here 6 genotypes are possible in the multiple allelism or blood grouping, right? Okay, please remember these genotypes of A, B, and A and B and AB and O blood groups, right? Okay, next let us see here blood transfusion. Okay, blood transfusion. Before that, blood group typing. Blood group typing. To detect blood groups, here anti-serums are used. Anti-serums are used to detect our blood groups. Okay. Here, see beta. Here, your blood sample is collected from an individual. Here, this is a slide. Okay. Here, this is another slide. Okay. Method of blood group typing. Here, for this sample, here sample is blood sample is collected on this slide. This slide is A and this slide is B. Here, this is the blood sample collected on this slide. Okay. So when anti-serum, anti-A serum is added to this blood, if clot is this blood group is considered as A group. If clot occurs in this B sample means when anti-B serum is added, so it is considered as AB, B blood group. Okay. See beta, clot. If clot occurred in
Sorry,
A,
can't hear your voice.
yes. Now it's okay. If here this is slide A, this is slide B. Okay, here this slide is added with the anti-serum. Anti-serum A. If clot is occurred, this blood group is called detected as A. If clot is formed in B, it is detected as B. If clot occurred on both, that is called AB. If no clot, that is considered as O group. Okay. So clotting A blood group. If clot occurred in the B sample or with the B antiserum, that is decided as B blood group. Here clot occurred in both samples, that is considered as AB blood group. If there is no clot, that is considered as O blood group. Okay. See. Here group A sample B means antiserum A, antiserum B. Okay. A group clot occurs, no clot. B blood group, no clot, clot will occurs. AB blood group, clot occurs in both. O group, no clot. Okay. In this way, blood groups will be detected. So this is called blood group typing. Blood group typing. Right? Okay. Blood group typing. Right? Please note down. Make it fast. Here, blood sample, if it forms clot with A antiserum, that is called A blood group. That will be decided as A blood group. If the clot occurred with the B antiserum, that group is considered as B blood group. If clot occurred in both samples, means with the A antiserum and B antiserum, so that blood group will be decided as AB blood group. If there is no clot, it will be O group. Okay, na? Very simple way, very simple method. Okay, so take a screenshot. Yes, Revati madam.
Good morning, sir.
Right. Okay, blood transfusion. Before transfer of blood, here two conditions are there. So one condition is here donor antigens and recipient antibodies will be taken into consideration. Okay, na? Blood for blood transfusion, here donor antigens and recipient antibodies will be taken into the consideration. Please remember, it is very, very important. Donor antigens will be taken into the consideration and recipient antibodies will be taken into the consideration. So this is the main important condition for blood transfusion, right? Donor antigens, recipient antibodies. Okay. Okay, now let us see who can donate blood to whom. Okay, see beta here, this is a box. A, D, A, B, and O. A, B, A, B, O. See, here donor. One more box. A group, B group, AB group, O group. Okay. Wait. A, B, AB, O. A, B, AB, O. Here, this person is donor, donors, recipients, recipient. Okay. A group. Donor A can give or donate blood to a person with A blood group. So here there is no clump will form. Clump means clot. There is no clump will form. But if A group person donate blood to B group person, here severe clump will form. See beta, clump means clot. Here this is no clot. Here severe clot. Severe clot causes harm. Okay. So A person can give AB person, but here slight clump will form, but it is not harmful. A person cannot donate O group person. See beta, here before this, see here A person. A person means antigens A is there, and antibody B antibodies are there. See, this is the symbol of B antibody. Okay, don't think as white. This is antibody. Okay, for example, this person donated his blood to B group person. B group person means here B antigens are there. And what kind of antibodies are there? A kind of antibodies are present in this person. Okay. So in this type of transfusion of blood, here antibodies, here recipient antibodies will be taken into the consideration, and the donor antigens will be taken into the consideration. See beta, here is recipient. Okay. So these antibodies forms clump with the A antigen. A antibodies forms clump with the A antigen of blood group A. Please observe seriously. Here a person, he is donor. Here donor is donating blood to a person with B blood group. Okay. In this donor, here antigens will be taken into the consideration. Here A blood group person is having A type of antigens and B type of antibodies in his plasma. Next, he is donating blood to the person with the B group. So in this type of transfusion, here this person has A type of antigen, B type of antigens, and A type of antibodies. So he is recipient, he is receiving blood from this person. So what kind of antibodies are there here in the recipient? A type of antibodies are there. So here these antibodies will make clot with A antigens. A type of antibodies makes clot or clump in the blood of A group person with A antigens. So that is why here this is not possible. Blood transfusion is not possible between A person and B person. Why? Because here severe clump will form. This clump may harm that person. Okay. In the case of AB blood group, AB group person, here easily you can take blood and a slight clump will form because here B antibodies are there and here A antibodies are there. Sorry, A antigens are there. These antigens, sorry, here A antibodies are there. Here these A antigens can make a clump with B antibodies. Okay, so that is why here small slight clump will form. It is not harmful this person. Okay, but here A group person cannot donate blood to O group because here A means A type of antigens are there in this person. A and B, both type of antibodies are there. In this person, A and B, both type of antibodies are there. So here, this A type of antigen can make a clump with A and B antibodies. So here, severe clump will form. It is not possible the blood transfusion between A person and O person. Right? Next here, B, this person cannot donate blood. Severe clump will form. Okay, no clump. Here, slight clump. Here, severe clump. AB cannot donate. Severe clump. B cannot donate. AB, okay, no clump. This person, severe clump. But O person, here no antigens. No antigens. That is why he can donate blood to A person. No clump. No clump. Okay. So in this way, we can study about the blood transfusions. Okay. Right. Please note down this. See what I hear. This means no clump. Slight clump. Severe clump. Okay.
So what is mean by clump?
What is meant by
Lump.
clump? Means clot.
Okay.
Okay, but I here in this table, let us see. This is blood genotype, phenotype, antigens, antibodies, donor, recipient. Okay. See what I here. Genotype. Genotype. Phenotype. Okay. Antigens present in this group. Antibodies. What kind of antibodies will be there? Next to donor, recipient. Means, okay, genotype, IA, I0. This type is called A group. Phenotype is A group. And this is blood group number 1. And the antigens, A type of antigens. Antibodies, B type of antibodies. And here can donate to A group and to AB group only. Can donate, okay, can donate transfusion. Donation is very simple word. Can donate to. Can receive. Can receive from. Okay. Can receive from. Can donate to. Okay. Can receive from means A and O. A and O. Is there any confusion? Okay, beta. Here, this person can donate his blood to A group persons and AB group persons. And this person can donate blood to, can receive blood from A group and O group. A and O groups. Next to second type of blood genotype, IB, 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 IO. Here this phenotype is B, means B blood group. What kind of antigens are there here? B type of antigens are there, and the A type of antibodies are there. And this person can donate blood to or transfuse blood to B persons and the person with AB blood group, right? Next, he can receive blood from B group and O group, B and O group. And here third one, AB blood group. IA, IB is the genotype of AB blood group. This person cannot, what kind of antigens? A and B type of antigens are present in the plasma membrane of the RBC, and antibodies in the plasma, blood plasma, nil. No antibodies. No antibodies. This person can donate blood to AB blood group only. AB blood group only. But this person can receive blood from A, B, AB, and O. From all types of people, he can receive blood. That is why he is called as universal recipient. Next, fourth one, IOR. Okay. Here, antigens. Here, this is O type of group, O blood group, phenotype O. Here, antigens absent. Antigens absent. Antibodies, A and B type of antibodies are present in the blood plasma. This person can donate his blood to A, B, AB, and O. Here, that is why he is considered as universal donor. Okay, but he can receive blood from only from O group. Okay, so this is very simple table beta, which explains about the genotypes as well as phenotypes and type of antigens and type of antibodies, and from which person he can receive blood and to which person he can give blood. So this table will explain about everything, all these things. Okay, please copy this.